So it's great to have Kate and Frank here together. I can't remember when I first met Kate, but the first time I did, I was sure impressed, and I'm not the only one. I'll come to the award she's won. But let me just say that first, she's in the School of Computing and Information Science. She's been the director of the director of the National Center for Geographic Information and Analysis at the University of Maine. Helped found it back in 1989, so it's been here a long time. One of the things that made me realize what a great asset she is to the university is kind of her training at the University of Wisconsin, which is kind of that beautiful mix between cutting-edge science and real-world problem solving. And I think a lot of people think that the University of Wisconsin is one of the land-grant institutions that does this work best of any in the country, and I often try to figure out whether we can catch up with them in any way. So she did her master's and PhDs there. She has won a number of awards. I'll just mention two. That last year she won the 2014 Career Recognition Award from Maine Advanced Rising Tide Center. Just a little bit about that, highlighting the significant accomplishments of women faculty at UMaine through their teaching, research, constituent service, and campus leadership. Sharing the professional successes of UMaine faculty raises the profile of women scientists and is intended to inspire colleagues of every rank. And I can definitely say I might not be able to represent all genders in this opinion, but she's an inspiration to many of us for her work. And then, as if that wasn't enough, she won last year the Presidential Research and Creative Achievement Award. She played roles in the Sustainability Solutions Initiative and has played really huge roles in our Safe Beaches and Shellfish project, the first project of the New England Sustainability Consortium. So it's a pleasure to have you here today. Thanks, Kate.
Thank you, David, for that nice introduction. I was sort of anticipating we were going to have a snow day today, but well, here we are. Yeah, I think we're the first one. I think the other two guys actually.
Well, last week we managed to get one in.
So we're almost 3% on snow days. So Frank and I are going to do a tag team on this presentation. I'm going to start out and give you a little overview of what we've been up to and controls here, and a little bit about where we're going. So start out with a little bit of background, and then Frank and I have been working, we started working with the DMR primarily, and their water quality data that they use to assess the water quality and safety of shellfish beds. And then Frank has just recently started working on the Maine Healthy Beach data. So we're going to look at some spatial and temporal patterns in this data, just to try and get a sense. I'll say a little bit more about it. It's a fairly large and complex data set, so we're trying to make some sense of this data. And then I'll turn it over to Frank, and he'll talk a little bit more about some of his more recent work, so the next steps where we're trying to go with some of this analysis. So this is a slide I actually borrowed from Cole, and she may have borrowed it from somebody too. So the key questions, one of the research questions that we're after, in a very broad sense, is so what are the drivers behind water quality, particularly bacterial contamination, either for beaches or for shellfish? And this kind of captures pretty much everything. Precipitation, which generates runoff, whether that be in an urban environment, an agricultural environment, forested, residential. So these are really a lot of the factors that are at play. And what we're really trying to do is tease some of these out in a little bit more detail. So again, we met with the DMR back in July and got their data, their water quality data. And this is the data that they use to determine whether shellfish beds need to be closed or prohibited. So they have a whole classification system of conditionally prohibited. And this was back in December, this posting. So if just a few days before this, we had heavy rainfall. So we had rainfall exceeding 2 inches. Pretty much all the beds. Well, it depends on whether it's just in the south, just the southern end beds closed, but kind of a blanket rule. In excess of 2 inches of rain in 24 hours, all the beds closed down. And the Maine Healthy Beaches, and this is sponsored by, so there's 2 different legislative groups here. The FDA is really responsible for the shellfish, and EPA is responsible for healthy beaches. So we're checking the water quality to determine if it's safe for swimming and recreational activities. So in both cases, we're kind of looking at their data and say, can we bring some new insights to what they're trying to determine about what's driving some of these factors. So let me start with the DMR data. Their water quality observations are in a stratified random sampling. And I'll show you they have several different growing areas that there are in their strata. And then within each growing area, they have a number of sampling stations. And there are 2,330 of these water quality sampling stations. And they're sampled about once monthly. They do extra sampling when they have a high rainfall, which they call a flooding situation. And so far we've just, the data goes from 1990 to up to today, actually. But we just got the data through 2013. So overall that's over 300,000 observations. And what they record is, well, they collect a water sample and they evaluate that for fecal coliform. At the same time, they also note what the tide level is, what the wind direction is, water temperature, they estimate salinity, and they also note what they call adversities. So these things would be things that might influence the sample. Was there precipitation, say 24 hours before they took the sample? Were there ducks or geese or boats? Some kind of wildlife, perhaps, near where they were taking the sample. So those are recorded as adversities. So this shows the growing areas. They're divided into the western and eastern part of the Maine coastline. And then within each of these, then there's the whole range of water quality sampling stations. Just a little bit about the Maine Healthy Beaches data. A little bit shorter time range. It goes from 2000 to 2013. And they're measuring a number of similar parameters. They record rainfall. If it's been in the past 24 hours, and they'll update it to 48 hours. Water temperature, salinity, and instead of fecal coliform, they specifically measure or they're growing and testing for enterococci bacteria. So that's one of the differences between the Healthy Beach data and the DMR's data. And so for the two, there's 2 different exceedance criteria. So in the case of the shellfish beds, each of the sample stations is tested, and then their classification is based on this exceedance criteria. The P9 has to be, if it's greater than 31, and this is counts of fecal coliform colonies, fecal coliform colonies per 100 milliliters of water, or a geometric mean of 14. And then in the case of the Maine Healthy Beach data, again, this is, so we're not so worried about ingesting shellfish here. So this would be if you're swimming, so this has a little bit higher threshold for the enterococci, 104 CFUs per 100 milliliter of water, with a geometric mean of 35. So these are kind of these these are kind of the thresholds that we've been working with to see where we have cases of impairment. So one of the first things we want to do is just kind of get a sense of spatial patterns in the data. So this is total exceedance frequency. That is how many times an observation exceeded these thresholds over the period for each of the growing areas. So this gives you a little bit of sense of which growing areas have some potential problems. I can point to, so this, I'm trying to remember this one. This one is, so WM, this is the Kennebec River, Sheepscot River, let's see, WS, this is Muscogee Bay, this one is WU, this is the St. George, and this one would be the Western, this one's the Fox Islands. So anyway, you can start to see that if some growing areas have more problems than others in terms of exceedance. So here we're showing the exceedance percent of observations that are exceeding this 31 CFU by station. So in this case, if it's showing as white, this means it's never exceeded this threshold level. So part of it, again, we're trying to see, again, what areas seem to be having problems and be a target of why might they be having some problems. And I don't know how many of you are sort of familiar with parts of the coastline of Maine. This is Idaho, depending on how you want to say it. These are the Fox Islands, let's see, Rockport, Camden, Rockland. Oops. In terms of some spatial patterns and sort of human influence, look at Idaho, which is hardly inhabited at all, almost never has a problem. If you look at the Fox Islands, this is Vinyl Haven, there's just more habitation there, and we see some more problems. The other thing you can sort of tease out from this is if you go higher up some of these into the river up into the estuaries, we see more problems than if you're on sort of on the open ocean. So this is again just looking at some general spatial patterns in terms of what we're seeing. We also want to look at temporal patterns, which is a little bit hard because of the nature of the sampling scheme. But a number of samples are all taken on one day, not synoptically. Some person has to drive around and get to all these stations, and in Maine you can imagine that that's a little bit challenging, how you might do that. So getting from here to here is a little bit not so straightforward. So this is Machias, and so all these observations were taken on the same day, and it's just sort of interesting to note that we can see, well, partly there's a spatial difference here. So we see there's problems with high levels in the Machias River. As we get out farther into the estuary, we see less of a problem. And the other thing is interesting, over the course of the day that these are taken, we have a change in tide level. So a number of these are on the low and ebb tide, while several of them are on the flood tide. So this creates a number of problems in terms of trying to analyze this data. I'm just trying to look at, again, what some of the factors are that are influencing this. So they collect salinity at the same time that they collect the water. Well, they determine the salinity from the water sample. And so this is showing the salinity levels on average for these water quality stations. And does anything sort of jump out at you with this? Anything look odd here? So red is showing high salinity. And we can sort of see that in most cases that it falls off. There's sort of a gradient that the salinity falls off as you go up the estuaries. Something wrong here? This is the Kennebec. So you can see there's just a large volume of fresh water coming down from the Kennebec. So on average, you can see that there's substantially much more freshwater input that we don't see in some of the other estuaries. So now, we're looking at again sort of the same location, and what you might notice is again there's and you sort of see this gradient that there's sort of on average we have less problems where we have higher salinity, but where we have a lot of freshwater inputs, it's a little bit more compromised on average. Okay, so one thing we want to do is, again, sort of see, again, on sort of a broad scale, do we see some spatial temporal patterns? So one thing we did was we applied this spatial temporal scan statistic to the fecal coliform observations that were greater than 31 CFU on an annual basis. So this is like saying if it exceeds this threshold, we can consider that an event. And do I have a spatial temporal clustering of events more frequently than I would expect to occur just by chance on a random basis? So what the scan statistic does, you could sort of imagine it like a cylinder that gets passed over the data set, and the footprint of the cylinder changes. So it goes from anywhere from small to fairly large. And then you can set this time. So this says are we looking for clusters that occur within a week, a month? So that can be specified. And again, so as this passes over, it's calculating a likelihood ratio statistic where there's a hypothesis that there's similar rates of occurrence both inside and outside the cylinder versus the alternative, which says that there's two different rates. There's a higher rate inside the cylinder versus outside the cylinder. So effectively what we get as a result is we would see a set of clusters of these events or exceedance observations more frequently than we would expect to occur by chance in these resulting clusters. So here's a run of this spatial temporal scan for the data for 2010. And so these are all significant clusters, and they're numbered here. So number 1 is here, and the dates are October to November. So you can see there's a time frame in there. These are associated with some time period. But again, what we're saying is that within those time periods, there was more exceedances than we would expect to occur by chance. So we looked at those, and on the bottom here are precipitation events in excess of 1 inch rainfall. And so for each of these clusters of exceedance events, we see sort of periods of rain. So we kind of knew this, that precipitation is a key driver, but this just sort of essentially confirms this is indeed the case, and it's one of the primary drivers. So when we see significant clusters, in general, it's associated with precipitation events, some sequence of precipitation events. And this is just a similar analysis for 2013. So once we looked at that, our next step was to say, so we know precipitation is a key driver. Can we tease out some more specifics in terms of characteristics of precipitation? Now there's this blanket rule, 2 inches or more in 24 hours, everything shuts down. Are different locations behaving differently under different rainfall events? So we want to look at some additional precipitation characteristics, the volume of the rainfall, duration, time since the storm, on what volume of storm it was. So things like the amount of precipitation in the previous 24 hours, 48 hours, and so on. problem is getting this information. So in the DMR water quality data set, I mentioned there's an adversity. And so they asked the person who's going out to do the data collection to record on their data collection sheet information about the adversity. And sometimes they do, and sometimes it's quite detailed in these cases. And in other times it's not, it just occurs as a P code. It just says P, there was precipitation, and you don't know if it was in the last 24 hours, if it was 1 inch, if it was a half an inch. So it's mixed, a little bit of a mixed bag. So we can't really rely on the adversity codes, and the comments are helpful. So what we're interested in doing is sort of collating the observation, the DMR observations, against the independently collected precipitation data. So there's multiple sources of this: National Weather Service, there's COCOBOS, which is the citizen science volunteers who collect precipitation data, and a number of others. And what's down in this corner here is looking at what do we have for a date, for one of our observation dates, what do we have for precipitation data corresponding to that date. So we have 9 observations of precipitation data. So that just gives the number of precipitation observations that we have for that date. And this is the number of DMR observations we have for that date. So it's trying to put these pieces together, which has been a little bit time consuming. And the precipitation observations are sparse spatially. Unfortunately, well, you can probably imagine where some of the points are, because probably in the center of those circles there. So this was interpolated, this sort of orange, red, yellow surface was an interpolation of the precipitation data for this date. We're trying to get an estimate of sort of what's the spatial pattern of the precipitation, so that we could attach it to the observations that were taken on that date. So that's June 3rd, we have June 4th. And we can see that again the pattern it's shifting, so we can't just say one storm has the same, is going to have the same effect in every location. This day, June 5th, and we see it shifting. It actually seems to be looks like it's shifting south, June 6th. So this is again sort of one of the stages where we currently are, is trying to assemble this precipitation data, so we can link that to the observations. Some other factors that we want to consider are sort of the human factors on the landscape. And this is just showing an example of an estuary where there's more development. And if we just look at these values, this is on average, we're tending to see more exceedances in this estuary than one that's forested, which we would expect. And so, I'll get to it in the next slide. So this slide is actually based on a coding scheme, and it's just sort of a preliminary coding scheme that we considered trying to get at the hydrodynamics of these individual stations. So are they at an open ocean? Are they within sort of in the core of the estuary, at the mouth of an estuary, the head of an estuary? Or are they at the mouth of a stream, in a stream channel, and by stream order? So this 21 would mean it's in a first order stream channel. This 41 would mean it's at the mouth of a first order stream. 46 would be at the mouth of a sixth order stream. Now we can start to see some perhaps what we're after is, again, not all locations are the same. So we can start to say, we expect some behavioral differences here. We can start to see it in this progression here, from first order to sixth order. The number of exceedance, on average, the number of exceedances is greater, which we'd expect because there's just a larger watershed behind this higher order streams. So some additional terrestrial factors that we would like to also consider. Again, some of these human factors. So it's a question of what are the reasonable parameters and what are the ones you can actually collect and get data on. So some of the things that we're considering are parcel density. State of Maine has a new addressing file, so all the residences that have official addresses have a point. So we're looking at sort of some density mapping to get a sense of what's the potential human influence along the coastline. Things like impervious surfaces. We also have information on overboard discharge sites, wastewater treatment outfall sites, and something that Sean and Brian are looking at is drainage density as another parameter. So that's sort of working. At this point, I'm going to turn it over to Frank to talk about this one.
Okay, we know the major drivers of precipitation, and while we're looking for other factors involved in this pollution process. And what I'm trying to show here is it's kind of like just try out, some modeling methods. I don't know how much you guys are familiar with artificial neural network, but definitely there are many modeling methods to doing this for detecting the major factors and down the road prediction of results. And right here, I just showing this try this artificial neural network. Just this one.
Top button has a little.
Point to.
Oh, you want to advance it? Is this using air?
Oh, okay. The data I'm gonna use kind of like a sub data set for whatever Dr. Beard mentioned before, these two data sets. And specifically for the DMR data, I just picked the sub data set of bacterial pollution events in response to storm. And this way just give you a little bit sense of how the spatial difference between the bacterial pollution or levels after storm. And sorry, which is the laser? Okay, that one. Actually, this is same location. Well, you first of all you can see the spatial difference, not over across the coast. Even one spot, even one location, there's a lot of difference between the bacterial response to the storm. For example, we here right here these means what? Pretty high bacterial. We call recovery rate. Means what? This bacteria count what?
New Hampshire says they can't hear Frank.
How about right now? Have to. Can you hear me? Hello? Hear me?
Yes.
Okay.
Okay. Sorry about that.
Maybe I have to.
I'm getting tags like.
The recovery rate, what we define here is bacterial count reduction in 24 hours. Okay. Here for example, after this, I use the subset of 2013 winters, one storm data. So after this storm, so this in the 24 hours, these reduce, I think, in about this category. Okay, you see, well, right here maybe a little lower. So this is like medium, something like that. Now we also
Frank, I'm still trying to make sure I understand your legend on the recovery rate in 24
recovery because each we call monitoring station or sampling site. We got every day to day, kind of like measurement for bacteria count, right? So, for example, first day maybe 200, second day 100. But because I think fresh water comes in, or can maybe ocean water comes in or come down, whatever, kind of like dilute whatever right here. So it may reduce these bacteria counts second days. Actually, this capability of the ocean or water body to kind of like recycling down the bacteria.
I think what I'm not quite sure I understand is which is the direction. What's higher recovery to reduce levels?
We
Is a low number there a greater
no low
potential?
number. This actually we have a different number called relative. This we call absolute rate. Well, we just put a name here. We not accurate, but now we call recovery rate. Well, maybe more directly, we just call reduction rate. Now, actually, right here now exactly reduction rate. 127 means first day for example 227. Now second day is 227. Then I call reduction rate is 127. So definitely going down. Okay. Excellent. So another piece of data right here show you the days to normal level. Okay. Normal level means based on the stress level. The Dr. Pierre mentioned before 31. How many days this spot need to reduce to 31 or less than 31? So for example, right here maybe from here you see only 0 to 1 day even. You see you can see any relation between this. Actually, this is exactly this spot. For example, if you got a higher recovery rate or reduction rate, you got a last day, you come back to normal. Right? Get it? So you can also see right here a little bit higher. It's also pretty short time here. Well, this right here maybe you have to consider initial level. You say this kind of like pretty low recovery rate. Right here also the battery pretty light. Also right here pretty also short time. But think about it, that's kind of by the river. So you have to consider initial level of bacterial count. Okay. So I don't know how much you guys know this model. Definitely Sean's here, but he knows. And this got some idea from natural neurons, right? Well, this is all this conception. So usually the neuron pick up a stimuli or send a signal through the axon and to synapse, then to the neighboring neurons. Okay, based on this idea, in artificial neuron, this is the model artificial neuron. This can mimic this whatever outside stimuli to put input the signal with different weights to neuron. Okay, then with different activation functions. Well, definitely this is pretty complicated. Well, maybe we don't know how many neurons involved to specific process. Well, right here only showing 2 layers and 2 neurons at each layer. But in reality, we don't know exactly. You just shown pretty complicated. If more, you see, more neurons involved in one layer, then you will have tangled, lines between them. Okay. Now back to our research questions. Okay, and this I made the conceptual network model I made before, and we know precipitation is a major drivers. Also, we will consider a lot of landscape metrics. Okay, through could it be based on watershed, and while so many process through here to output bacterial pollution level, well, even you can consider non-point pollution source, point pollution source. We don't know. So many researches or publications down there discuss about even longevity of bacterial or pattern for runoff or transport from from peak mountain to the estuary, eventually to the ocean. Or, well, sometimes you will also have kind of non-stream. Well, mostly this one should be related to stream, okay, through certain channel. If non-stream event, for example, kind of like Dr. Fuyu mentioned before in DMR data, kind of, for example, one boat passed by, contaminated to the water body, something like that. This kind of we call non-stream event. Now, eventually, this output right here is only one output bacterial level, or we consider threshold level here. These pretty complicated problems we can simplify it right here. Okay, we can consider whatever known factors, whatever parameters available with data, already. We can consider these kind of things as input. Well, definitely lots of unknown factors and relationships between these factors. Or maybe partially known, so what doesn't matter. You can consider this black box or gray box we discussed before. And right here, now output here is indicated bacteria level. In DMR data, we consider fecal coliform. In beach data, we use enterococci bacterial. This is pretty simple model, and but with one hidden layer, okay. But everything we start simple. If a simple model can solve your problem, why try more complicated, right? But anyway, right here, this is just try out what's going on. Actually, for example, right here, but these whatever the weight, you see, only this simple model, you can a lot of line tangle, which different weight could be different weight, okay, and output also can make different weights, okay. If you consider 2 layers, okay, you can input neurons, output neurons, eventually output to the output makes it too complicated. And so I just try out one data set. This one's 2012 data in response to the floods. I think about it from the spring, summer, fall season. Also, they have some storm events occurred. I just put all of them here. So, right here, the MSE, the mean square error, is used to control monitoring this learning process, or called training process. Usually when we got the data set, we can split different percentage of a data set. Oh, for example, right here, I use called 70%. Okay, 70% of data used for training. okay? And use a 15%. Well, you can manipulate a little bit, and I use 15% of the data for cross validation. Cross validation during the training process, you also trying to check see how the MSE to satisfy whatever we preset the criteria. Okay. From here, so the pretty, based on the model, we got pretty low, MSE. Okay, this will tell you the modeling process or training process stuff. And so these slides showing the result of remaining 15% of the data set. Actually, it's independent of whatever training process. Okay, and so this one about a little bit more over 200 data points. So this output you can see how many points match. So score output means what? Means the output or value from the models. Okay, the score right here means actual measurement or observation, and these are some statistical criteria to measure this. Well, you can see, but the correlation coefficient is only 0.5. Okay, you can low. Actually, not that bad. Think about it. So how many factors you don't know this process? I will show you next slide. How many factors I consider right here? Okay, 0.45 is not that bad, right? And you see these, even months, latitude, longitude, all growing areas, plus tide stage, wind directions, and temperature, water temperature, the salinities, so many factors. So you can see right here. These are not set of output from these artificial the neural network models, because sensitivity analysis showing the relative significance of each input factors. So you will see the relative significance of these factors. Next, I don't want to explain all the factors. I just picked a summary for a factor here. For example, this growing area, okay, WV, W. Well, you can interpret this, okay, maybe higher value of bacteria, or some other, we don't know, maybe closer to the river or whatever, adversity factors could be. Okay. Anyway, this showing the spatial difference in this model process. Also, for example, these wind directions are pretty high. This is a southwest direction, also pretty got high weight. Well, actually, right here, by our intuition, kind of makes sense. So, look at the Maine coast. If southwest winds, kind of like a blow off whatever contaminated stuff on the land, right, to the ocean. It makes sense, but we don't know exactly the mechanism behind. And so this is stage. This is test stage. Is the test stage high? Well, this also weight. Is the salinity, the temperature is about pretty high weight. Well, if you only look at this number here, you don't know, okay, is a negative impact or positive impact. If you combine with this kind of graph, you will know, okay, this is positive impact or negative impact, right? This, for example, right here, I just pick up two. Okay, this is temperature. So with the increase of temperature, so this bacteria level go up. That makes sense. Kind of like bacteria is more not too high, but within the certain region. But definitely with the increase of temperature, bacteria grow faster. Okay, kind of like more active the cell, bacterial cells. And also salinities here. So increase in salinity will reduce the bacterial level. It makes sense. Actually, this is consistent to the Dr. Beard showed you along the distribution, the salinity, and the bacterial, the distribution, right? It's consistent. And well, I don't know how many people you can watch it here, but this is a little bit weird. Okay, the salinity is that high. No, this is after I finish this analysis. Usually salinity about I think even normal ocean kind of like about 36, 37, the most kind of like right here. No, I'm definitely if a lot of fresh water input definitely will reduce the salinity. Right here, these are really kind of like a normal, okay, data. So after I got this one, so this is I found this kind of like could be typo in there, but this data in the data set, but this definitely is maybe not available data, something like that, okay. But the trend should be the same. So I also try out some analysis of Maine beach data, on Enterococci bacteria. And similarly, we applied methods in DMR data set, but based on the thresh level of 104, and but you can give it some, first impression of how safe, how healthy the beach is, or not. For example, this East End Beach is by Portland, right? I don't know how yeah, this should be this is kind of like by big city. Okay, this makes sense. So this is pretty high, but think about this is about 14 years data. Okay, here, this 62 means in 60 times measure exceeding the threshold in 14 years for this spot sampling spot. Well, if you look at down this group, maybe okay, we can consider this beach much healthier. So only in 40 years, only 1-2 times, got exceeded in terms of level. So actually, this accidents frequency give you kind of like some ideas about this bacterial function. And also, I use scan statistic, which Dr. Beard introduced before. And right here, right here, only difference here, I've used a DMR. I analyze, just based on annual data. Right here, I just trying to use all 14 years. Because this initial analysis, I try to kind of see how this is a course analysis to see what kind of pattern or clusters we can find, based on year to year. Okay, and also you can see, okay, you can find hotspots. Can find from 1 to 5, 1, 2, 3, 4, 5. But these are pretty small. But only I will show you the actual data in next slide. Right here also includes the accidents frequency here. Well, you might see here a little bit weird. It's well, this is pretty high. Why not even cluster? Why? Yeah, it's different because with two variables to consider here. We choose this is a space and a time. Consider also this could be the frequency could be dependent distribution, but in 14 years, if not a cluster, if sparsely distributed along the 14 years, so you still couldn't catch this pattern. Well, we not consider, but if for the further analysis, if year to year, maybe we can catch some kind of pattern for some years. Also, we can correlate it to this kind of pattern to precipitation or other factors. Okay, this now just gives some roughly the course patterns right here. So these patterns, these results, specifically describe to whatever the pattern I shown before. And for example, first clusters include what kind, how many sampling sites and at a specific beach in time frame, or what kind of radius, what kind of how expand for this hotspot. Okay, I don't want to describe all of them, but anyway, this is patterns. So similarly, I also try out some artificial neural network modeling to these 14 years data sets to see how they're going. Okay, is similarly use as MSE to monitoring the training process. Okay, so actually this process means as long as you can see, okay, because these are two different data sets. But first you use training data set to training the models. Then use average training process and use a cross validation data set to check to see how the mean square error. So eventually all going down means that this model are pretty good, at least at this moment. And this one also use independent data sets or rest of 15% of data in trying to predict how these models give the results. So actually, these one give pretty better output. You see the correlation coefficient is about 0.8. Okay, well, definitely we need to further refine this model. Well, because in so many factors, right, used here could be there could be some redundant, factors here. So these are preliminary, are not final. And similarly, I apply, use the sensitivity analysis to. Why I use I think the first slide I use right here use 40 precipitation of 48 hours. These data I don't know these data sets kind of like two parts. From 2000 to 2007, they only recorded 24 hours precipitation accumulation, and after from 2008 to 2013, they use 48 hours accumulation. So I just use the second subset to run these models. And from this sensitivity analysis, you also can show you the relative significance of salinity in these models. Okay, it's pretty high. So next steps. Actually, right, I showed before only kind of like bacterial level. Definitely, we can use this modeling or other spatial analysis method to the recovery rate. Okay, we also can model this one. We already can from first, kind of even from frequency or just distribution of these accident frequency. You can see some difference between across the main coast. Definitely, this is actually recovery may be more significant to even stakeholders also interested. And we definitely in the future definitely we're gonna do more kind of flat data evaluation or analysis and incorporate more precipitation or especially storm characteristics and some landscape metrics. We can collaborate with other teams members to design more denser time points. So maybe during the summer or during the fall season, we can design a new experiments. Based on whatever experimental design we can use, and just incorporate more parameters from or terrestrial parameters. And similarly, we can use recovery times as dependent. Definitely, how reliable well, we can use different models. So definitely, we also can try this artificial neural network or use some other methods. So this could be very important to output recovery rate and recovery times. Any questions about me and Dr. Beard?
So we're right at time. If people have to excuse themselves, go right ahead, but there's time for questions too, so please speak up.
Wow.
I just want not a question but a comment.
Okay.
Bravo, because I know both of you have taken on a lot, so it's wonderful to see you keep taking on more and more. Thank you.
Thank you, guys. Thank you, Dr. Beard. Good guidance.
What great teamwork. I was wondering with the artificial neural net results, do you have any sense? I mean, I was pretty impressed, particularly with the beach data, with the results you were getting. How much, where else has this been done for this kind of a problem, and how does what you're finding compare with the ability to use ANN in other settings?
This good. That's good. Very, very, very good. Because this, but definitely, we are in a computer school, computer science, right? Definitely, we can pinpoint the results, use different models. Actually, different functions within artificial neural network. How huge functions. Because a lot of unknown process right here, you don't know if it's a linear relationship, you don't know if it's a nonlinear relationship. Definitely, we can prove one by one. I don't know. Maybe for a time frame, maybe 2 years, 3 years, I don't know. So that's why I request funding for the software, which kind of speed up, expedite our process. We can, because I use trial version, because this is limited functionality. I can try just a couple of very simple functions. With the commercial professional versions, I can do much more stuff. You could provide more promising outputs.
So most of the studies that I saw, they're really just using regression.
But they try different topology. They call different try because because a lot of too complicated. You don't know how different layer. Why they call neuron? Because human beings, neurons, they kind of like even you see, for example, after 15 years, you know the friends, you still recognize the friends, and before 15 years, hey, still ready because this kind of fuzzy process. So neural net kind of like trying to mimic that process. You see huge data, lots of abnormal stuff there. So I think based on the model I shown here, kind of like these artificial neurons quite fit whatever we trying to do right here for prediction or finding other potential major factors.
Yeah.
Is it possible to ask a question from over here?
Sure.
Yeah, go for it.
Nice talk. I was curious on for your cross-validation, your airmet disease, what was that relative to? What was the mean of your training data sets for both data sets? So for example, for the first one, like you had a mean absolute error of like 44 CFU.
Yeah.
And for the second one, it was 213. And so I was just curious, what is that relative to? What was the mean of your original training data set? Just for an idea of how well that looks.
I think in reality, it's kind of like when you use the artificial neural network, it depends on how huge the data set you have. That then decide what kind of percentage for each category. And for my case, that's I said some people suggest kind of 65% for actually based on percentage for your the data you have. For first step, for example, right here, first data I have a kind of like 14, I think the 14,000 data, I use 70, 15, 15. 70% use as a training, 15 use
Right.
cross-validation, 15 use independent testing.
Sure.
So similarly, users apply to the second data set. But this I just tried to the preliminary results, not the final result I tried to present in my publication. But I would definitely, we're gonna incorporate more relevant terrestrial data or factors.
So I think just in the interest of time, hang on a sec. I think some people do need to head off. Totally understandable. And for those who want to hang on, let's continue this conversation. And I know you had a question too. So why don't we give people a chance to break and thank Frank again and Kate for a great presentation. Was there more UNH that you guys wanted to ask?
I'm still here, so any questions?
Oh, I have a question like, is it possible to use your model to decrease the amount of input?
Oh, definitely. These are
Identify
pretty
what
flexible.
is important.
Well, actually, ideally, you see, run the first one round, second round, then you will see some factors are not that important. So you just remove them. So then you remove them, you just reconstruct your data set, run second round, then see how that, could probably give you better output results. So you keep running. Depend on scientifically or methodologically, you see if you can, interpret properly your data. Japan, I think it depends. Definitely, you have to run one round, second round, then remove redundant stuff.
Yeah, and both your talks never mentioned the condition of the ocean forcing, such as waves and circulations. Because let's say you have same amount of pollution come from the rivers to the beach. If the beach has strong current circulation waves, the pollution going to be mixed, dispersed, carry away faster.
Definitely, that's why the fact that's why we last dependent variable, recovery rate and recovery time should be different, space to space. Okay, but the power and flexibility of this methodology is here. It's a new artificial neural network. You don't have to consider the specific. Otherwise, if you can pick specific experimental sites, definitely you can back to traditional spatial analysis methods. Definitely you can use that, or use kind of like traditional statistical method. But the beauty is here, this modeling, you don't have to consider that. You just put in the data, kind of like now even Google search. Now Google search use a lot of kind of like this machine learning for that. That includes this artificial neural network. They just based on whatever data you provide, then just run the learning process, just like mimic human beings. See if you can find some kind of patterns in your data.
But your model only apply for particular site.
No, you don't have to. No.
Because different sites have different forcing. For example, if it's ocean forcing is different and have different dispersion effect on your same
If you put that data, the model will tell you, okay, this is different.
Yeah, but what happens if you include some ocean forcing in your model?
Definitely could be, because all these data are along the coast. No data is along, on the land. So it should be all that ocean occurrence should be affect. That's a good factor actually.